Alright, so we're going to start up with our next section talking about proteins, and before we get into the structure of proteins and review a little bit more about dehydration synthesis and hydrolysis, which like for lipids, carbs, and nucleic acids are how we build proteins, we want to look at the two main categories of proteins and what they do. Alright, so for some proteins, they are structural. The proteins that make up your fingernails, the proteins in your skin, the proteins in your muscle fibers, the proteins in your hair, those are all structural proteins, as opposed to enzymes, which are more functional proteins that are going to carry out the job of catalyzing chemical reactions. And so what that means is that for some reactions in your body, they happen really, really slow. But if you had a catalyst, they can accelerate by literally hundreds of millions of times. And we'll see some examples of that. And we'll have a whole chapter on that here coming up very shortly. Chapter 4 will be like that. Now for proteins, as compared to carbs, lipids, and nucleic acids, they are very, very abundant. If you were to pull all the water out of a cell, roughly half of what you'd have left would be protein biomass. Now, in the world of proteins, the protein itself is the macromolecule, it's the polymer, and the building blocks or the monomers would be the amino acids. So like where polysaccharides are built using monosaccharides, and where nucleic acids are built using nucleotides, and where lipids are built using fatty acids, now we have proteins being built of amino acids. And so just like with any of those other categories, we're going to be talking about dehydration synthesis. Now, in the case of proteins, and this is going to be kind of similar to what we're going to see with nucleic acids, it's the sequence of the building blocks that makes a difference. So the sequence of the amino acids is kind of like the sequence of letters in a word. If you change up the sequence, you change up the meaning. All right, so it's really, really important to make sure that you have the right sequence of amino acids in order to have the proper sequence in the protein, so that the protein has the proper function. Okay. Now, dehydration synthesis we've talked about before. We're going to talk about it again. If we remove a molecule of water, if we take an OH group from one amino acid and an H from another amino acid, well, that's two H's and one O, which of course is water. It's H2O. So that's how we're going to create a covalent bond between adjacent amino acids. Right? Now, with proteins, one of the other functions or ideas that we're going to see with proteins is that the shape is absolutely critical. More than any other category of macromolecules, the shape is very, very important. If you change the shape of a protein, you change the function of a protein, and that's a really important thing to remember. So what determines the actual shape of the protein then is the sequence of amino acids, like I mentioned a second ago. What determines the sequence of amino acids is the sequence of nucleotides in DNA. So DNA is the recipe that we use to build proteins by determining the sequence of amino acids, therefore the shape of the protein. Right? And that's basically what this entire course is about. If you change the DNA, you change the sequence of amino acids, you change the shape of the protein, you change the function of a protein. And we cannot say that enough. Okay, so protein shape is everything. It determines function. It's really important to remember that. All right, so moving on, we're going to look at how these are actually categorized in different groups. We talked about different types of structural proteins. Keratin would be one, like we see in this hair, this dog up here, or in the horns of this bighorn sheep. The outer covering over the bone is actually made of keratin, which is the same stuff in your hair and fingernails, toenails, versus spiderweb silk, which is also very, very strong. It's also a structural protein. One of the strongest proteins we have actually discovered is spider silk protein, so it's a fairly impressive one that way. Right, so proteins, again, they are polymers, and they are built out of monomers, like we've seen in our other categories. In this case, we have amino acids strung together to make long chains of what are called polypeptides or proteins. Polypeptide and protein is basically the same thing. Right, so the reason why we call it a polypeptide is the bonds between amino acids are called peptide bonds. So if you have more than one peptide bond, you therefore have a polypeptide. So the name really kind of makes sense. Certain kinds of proteins have other things attached as well, so you can have glycoproteins where you have a carbohydrate attached to a protein. You can have lipoproteins where you have a lipid group attached to a protein. So those other groups give the proteins a different function than if it was just a protein by itself. So we'll see those a little bit as well. Good examples of lipoproteins would be HDL and LDL, which maybe you've heard of before. These are your good and bad cholesterol, respectively. Together they make up your total cholesterol, so they actually represent different categories of proteins with lipid groups attached to them. Glycoproteins we see a lot with cell receptors. On the outside of cells, there can be receptors which can grab hormone messages or other kinds of molecules that might be floating by in the outside environment. Some hormones also are glycoproteins, and you see those maybe in a future course when you take A&P. And again, amino acids are the monomers that are used to build the polymers of course being the proteins or polypeptides if you like that name better. All right, so if we look at the structure of amino acids, they look like this. Carbon's front and center. This is another one of our organic molecules. So we have carbon, and as we've talked about before, carbon likes to form up to 4 bonds, and only 4 bonds, right? So the first bond that carbon forms is with an amino group. NH2 is an amino group, so that becomes the amino part of the amino acid. On the other side, we have what's called a carboxyl group, otherwise known as carboxylic acid. So on the other side, we have a carboxyl group. That's the acid part of the amino acid. Right? We also have a hydrogen molecule, or atom attached to the carbon as well. And then the other group up here is a variable group, and so we call that an R group. Right? So for all of the 20 amino acids, and there are 20 of them, they all have this structure. The amino right here, the hydrogen down here, the carboxyl right here, and then carbon right in the middle. Right? So what makes those 20 different amino acids different is what appears up here where this box is. So depending on what that R group is, that's going to determine the function of the amino acid. That determines what effect that amino acid has on that particular part of a protein as far as its shape. So sometimes the R group might be polar, sometimes it might be nonpolar. That, of course, can have a huge implication when it comes to how that amino acid functions and how that particular part of the protein functions as well. If the amino acid is cysteine, which is one of the 20, the R group actually has a sulfur attached to it. And different cysteine molecules, when you have them as part of amino acids, give the ability to form what are called disulfide bridges, or little cross links in the overall protein structure. The sulfurs bond together and form what's called a disulfide bridge. If the R group is just a hydrogen atom, then this molecule becomes glycine. So glycine is the simplest of the amino acids. This R group would actually just be an H. Otherwise, it would be some other group of atoms together to form the R group, depending on which amino acid you're looking at. So R is kind of like a variable, right? The R is an unknown depending on which amino acid you're looking at. If there's a sulfur up here and some other atoms, it's cysteine. If it's an H, it's glycine. If it's some other stuff, then it's some other amino acid, right? So we've seen this a bunch of times before. We're going to look at it again. Dehydration synthesis, right? So we have an amino acid over here on the left, amino group, hydrogen, carboxyl, R group, carbon in the middle. We have amino acid over here, amino group, hydrogen, carboxyl, carbon in the middle, R group up there. It doesn't really matter what the R groups are for the case in the case of this example. Okay, so if we take out this OH and this H, that gives us H2O, and that creates a need for a bond between this carbon and this nitrogen. We've taken one bond away from the N, one bond away from the C. They both want to have another bond, so they're going to bond together. Carbon bonds to nitrogen, and you get what's called a peptide bond. The bond between what used to be a carboxyl group and what used to be an amino group, this bond right here is a peptide bond. And so if you have a bunch of those, hundreds, thousands, tens of thousands of those together in one molecule, you have a polypeptide. And again, cannot emphasize this enough. The specific sequence of amino acids determines the shape and therefore the function of the protein. Okay, so that's our basics on proteins. Now, you might wonder, why or how can 20 different amino acids make all the different proteins we see in every living thing anywhere? It doesn't matter if you're a bacterium, a tree, a protist, algae kind of a thing, a fungus, an animal. Humans are an example of that, right? So it doesn't matter what kind of organism you're looking at. We all use the same 20 different amino acids. And so the way this works is the same way that we can have different words formed in an alphabet. So in the English alphabet, we have 26 letters. And by rearranging those 26 letters in different lengths and different orders, we can make lots and lots of different words, something like 450,000 words in the English language. Right? So we don't need to have 450,000 letters. We can use the 26 letters, change up the order, change up the length of the word, and get all the different words that we need with all the different meanings. And proteins really work the same way. There are 20 different letters, amino acids in this case, which are strung together in different orders, different sequences, which gives you different proteins with different functions. Okay, so for this little protein down here on the bottom of the screen, this would be a very simple protein. You've got the end amino group over here. You've got glycine, isoleucine, valine, glutamate, glycine, cysteine, alanine, serine, and so on. These are all just different amino acids. And the only thing different about each one of these is that R group that we talked about before. So if you were to change out one of these amino acids for a different amino acid, that would create a very different protein, potentially. Not always, but potentially that might create a very important difference in the protein, which could affect its function. Right? So if we look at the structure of proteins, something like this. The primary structure of a protein is the actual just order of amino acids. This picture back here, this is the primary structure of this protein. It doesn't show you anything else about how it folds up, doesn't show you any loops or bends or hairpin turns or spirals or anything like that. It just shows you a sequence of amino acids. That is the primary structure of a protein. The most fundamental structure is the primary structure. Secondary structure is how those amino acids would begin to interact with each other. So hydrophilic ones would kind of go to one side, kind of try to orient together. Hydrophobic ones would try to orient together as well. And so when they start to do that, they create secondary type features like a helix or like what's called a beta pleated sheet, sort of a zigzag pattern that you see in some proteins as well. So that's another option that you might see in some proteins. From there, we go to what's called tertiary structure. If we take all those secondary features collectively and consider how they affect the shape, that creates a tertiary structure of a protein. Right, so for some proteins, they only have a single chain of amino acids. And if that's the case, tertiary structure is as far as you go. Some proteins, though, like hemoglobin, for example, which is the protein in our red blood cells that helps to carry oxygen and to a lesser degree CO2, hemoglobin is actually made up of 4 amino acid chains. That polypeptide is made up of 4 separate chains of amino acids. Each amino acid chain has a primary, secondary, tertiary structure, and when you take all of them together, you create what's called a quaternary structure. Right, so for quaternary structure, that's all the polypeptide chains together. Tertiary is within a single chain. You see the disulfide bridges we talked about before with cysteine. We see hydrophobic and hydrophilic things lining up. And again, for a protein with only one chain of amino acids, that's it. But if you have a multi-chain protein, a protein made up of multiple chains of amino acids, then you can also go to a 4th level of structure like we see with hemoglobin. All right, so for an example up here, we see again primary structure, some generic amino acids, amino group, R group. The hydrogen would have been right here originally. Actually, it's right here. This would have been a carboxyl group originally. This is the R group, and you connect this one to this one, to the next one, to the next one, to the next one, and so on, each time by a peptide bond to create a polypeptide molecule. This sequence of amino acids is primary structure. When they start to interact to form things like this helix or these beta sheets, that's secondary structure. All those secondary features together makes up tertiary structure. And then again, only for proteins that have more than one chain, you start to consider quaternary structure, where you've got the different chains interacting among each other, among themselves. Right? So the actual amino acids in their sequence, the way they fold up, creates the function of the protein. Now, there are cases where that structure of a protein can begin to break down. Normally, these are fairly well stabilized. Some proteins are more stable than others, but for the most part, these proteins are fairly stable. If you start to break some of these stabilizing forces, like the covalent bonds, the ionic bonds, hydrogen bonds also form within and among different polypeptide chains, you start getting into what's called denaturation of proteins. A denatured protein is a protein that has unfolded itself. The bonds that were in place are no longer present, and the protein begins to unfold itself. It would be a bit like a sweater that you've got a loose thread on it or string on it, you start to pull it, and before you know it, the whole thing is starting to unravel. And if you continue to pull, you just have a big pile of string at the end. You've taken what used to have a shape and you've unraveled it, and now you have just one continuous thread with no real rhyme or reason to how it's folded up. That's really kind of how a protein works as well. If you denature a protein, you unravel it, you still have primary structure potentially, you still have your sequence of amino acids, but all those secondary, tertiary, maybe quaternary structures have come apart, and now the protein doesn't work anymore. Right, so there are several features which can cause denaturation. One of them is pH. Most proteins have a range of pH in which they like to operate. So for example, blood proteins like to be in a range of around 7.3 to 7.4 pH. Stomach proteins like to be in a range of around 2 to 3. And if you take those proteins out of their natural environment when it comes to pH, they begin to denature. If you take a stomach protein and put it in the blood, or a blood protein and put it in the stomach, neither one of those proteins would survive. They would both begin to denature. And this is also the basis for how pickling works. Back in the old days, before refrigeration was common, they would take fruits and vegetables from the garden, they would put them in acetic acid, which is vinegar, maybe some spices in there as well, and that low pH vinegar would preserve the food by preventing bacterial growth. So what causes food spoilage then is bacteria. If you create a hostile environment for those bacterial enzymes, those bacteria can't grow anymore, and your food stays good for maybe several years. And so pickling was just based on using low pH to prevent bacterial growth. Temperature also causes denaturation. So if you take an egg, crack it open, dump it on a hot frying pan, what's the first thing you notice is that the egg white, which was initially clear, that's albumin protein, turns white. And that white changing color of that protein indicates a denaturation of the protein which has taken place. Once it unfolds, it's not going back to being folded again. You could sit there for the rest of your life and wait. That egg white is never going to go back to being clear or transparent again. It's going to stay white forever. Okay, so the denatured protein doesn't go back to being folded up, just like your unraveled sweater wouldn't go back to being folded up on its own either. right? So temperature will also denature proteins. The reason why cooking food thoroughly is so important is that any bacteria that might be growing in that food will have their enzymes denatured by the high heat. That will prevent them from growing and make the food safe to eat. You can also change the ionic concentration. Basically what that means is salt. And by salting the food, that also prevents bacterial growth, right? So by creating a hostile ionic environment, that prevents bacterial growth. And again, that makes food safe to eat. So back again in the old days, before refrigeration was super common, they would have salted meat, for example, and that salted meat would stay relatively edible for a long time because bacteria couldn't grow on it and couldn't make it spoil, right? So that's another way that we can cause denaturation. So that's going to take us into our last section of macromolecules, which are the nucleic acids. Nucleic acids are primarily for our purposes the ones that we're going to look at: DNA, RNA, ATP, and a little bit on a couple called NAD+ and FAD, which we'll see later on in a chapter on cellular respiration. So starting off primarily with DNA and RNA, which is a lot of what this course is about, these are the molecules of heredity. These are the molecules that cells use as information storage. So if cells divide, they pass on information about themselves to daughter cells using DNA and RNA. Other kinds of nucleotides can be used as messenger molecules. They can be used as energy storage molecules like ATP. They can be coenzymes. The ones we're going to talk about the most are DNA and RNA. Okay, so in this case, nucleic acids are the polymers. The monomers are what are called nucleotides. Nucleotides are the building blocks that you need to make nucleic acids. So in terms of nucleotides, the overall makeup of a nucleotide is actually three things. You have to have some type of 5-carbon sugar, you have to have a phosphate group, and you have to have a nitrogenous base. This means a molecule that contains nitrogen. All right, so if we look at these different types of nucleotides, there's kind of two main types if we're talking about DNA and RNA. If we're looking at the sugar involved, DNA has deoxyribose as the sugar, and RNA has ribose as the sugar. Okay, so that's one big difference between DNA and RNA, is the 5-carbon sugar present. Deoxyribose for deoxyribonucleic acid, which is DNA, or ribose for ribonucleic acid, which is RNA. We can look at the different bases involved as well, right? So there are two bases classified as what are called purines. Those would be adenine and guanine. And there's actually three bases classified as pyrimidines. These are thymine, cytosine, and uracil. Okay, so depending on which one you're looking at, DNA versus RNA, DNA, for example, is going to have A, C, G, and T. Adenine, guanine, thymine, cytosine. RNA is going to have A, C, G, and U. Adenine, guanine, cytosine, and uracil. So again, the bases differ also between DNA and RNA. And the bonds between adjacent nucleotides, in the case of nucleic acids, are called phosphodiester bonds. Just like proteins have their own special bonds between amino acids, peptide bonds, nucleic acids have special bonds between nucleotides. We call those phosphodiester bonds. Okay, so we'll see those too. This is a visual representation of a single nucleotide. This molecule down here is the 5-carbon sugar. If this blank space here is an OH, it's RNA. If the blank space is just an H, it's DNA, hence the name deoxyribonucleic acid. DNA has one less oxygen than RNA has in its sugar. There's a phosphate group up here, and there's also a nitrogenous base up here as well. Now this phosphate group, one important thing to notice about this would be these negative charges here. And we're going to come back to those later on and talk about what implications those negative charges have for the molecule in question as well. Right, so this picture is fairly complex, but it's showing you, again, kind of the pieces here. There's three things going on here. Up here is a single nucleotide, phosphate group, sugar, in this case deoxyribose because this is an H, and a base, which in this case is guanine. These 5 pictures here show you the 5 different nitrogenous bases. And again, obviously we're not going to ask you to remember the structure of these for the purposes of this course, but that would be A, C, G, T, and U. So again, DNA is A, C, G, T, RNA is A, C, G, and U is its 4th base. Down here, they're just showing you, again, dehydration synthesis, the same reaction we've seen before, to create a polymer of nucleotides, which collectively then would be a nucleic acid. So from this sugar up here, we take an H. From this phosphate, we take an OH. That creates a phosphodiester bond between this nucleotide on top and this nucleotide on the bottom. Now we have a nucleic acid, very small nucleic acid, but still a nucleic acid nonetheless, that's made out of 2 different nucleotides. As far as DNA versus RNA, another big difference between them is the number of strands involved. So in RNA, we have a single-stranded molecule, and for DNA, we have a double-stranded molecule. And again, these are very complex, but also very repetitive. So what we see up here, a base, a sugar, a phosphate, a base, a sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate, repeated literally tens, hundreds of millions of times for a single molecule. And we'll notice in DNA how there's these two strands of nucleotides together that are joined in the middle by these dotted lines. And we've seen dotted lines before represented as bonds. These are actually hydrogen bonds. So we'll get back to those later on and talk about why they're important, but really the main reason is the two strands of nucleotides are held together in the middle by hydrogen bonds. They're very, very critical for that reason. All right, so I mentioned before how in proteins, the sequence determines the function. The same thing is true in nucleic acids. The function of the nucleic acid is determined by the sequence of bases. Notice how all the nucleotides have, really 2/3 of them are the same. They all have the same phosphate and the same deoxyribose in this case. Same phosphate, same deoxyribose, phosphate, deoxyribose. These are all the same. The part that's different is these bases. And so if you change up the sequence of these bases, that's going to change up the meaning of that particular molecule of, in this case, DNA. If you change this T to a G, this becomes a C, and everything changes. So it's very critical to have the right sequence of bases for that particular chunk of DNA to work the way that it's supposed to. And again, like I mentioned before, they're connected in the middle by those hydrogen bonds. We also have what are called base pairing rules, which says that in two strands of DNA, thymine always goes with adenine, and cytosine always goes with guanine. A always goes with T, G always goes with C. It always works that way in the case of DNA, at least it's set up properly. Right? So we have these hydrogen bonds between adjacent, what we call complementary bases. A and T are complementary, C and G are also complementary because they bond together. All right, so we're going to cut off here, and we'll make one final last section on this chapter.